We're talking about Spirometry Lab, a quick review by me and Panshul. First one question is, what is dead space? Can someone answer what is dead space?
The space which ventilation doesn't happen, doesn't occur.
A place where what?
Ventilation, the
Yes.
gas exchange.
Oh, that's good.
Yeah.
Really good. Okay, so basically, first, to answer what is dead space, we need to know the difference between shunt and dead space. Do you know the difference between shunt and dead space? Someone? Was that Kirari who answered before?
Yes, it was me.
So what's the difference between shunt and dead space?
I think shunt, it's something related to vasculature.
Yes. So you said dead space was what? Absence of?
Where the ventilation doesn't occur.
Yes, and the shunt is absence of?
Of vasculature.
Yes. Yes. So basically that's it. The shunt is when you have absence of air. So for example, there is a block here. So here there is no air. This will be called a shunt. And the dead space is something that there is the air is coming in, but there is blockage of the blood, so there is no blood coming in. Okay, so that's the dead space. Dead space is basically there is air, but there is no blood. And shunt, there is blood coming in, but there is no air. Okay, so these two terms you should remember. This is the normal. It's supposed to be like this. Blood is coming in, air is coming in, that's the normal. If you don't have the air, because there's a blockage there, it's called a shunt. And if you don't have the blood, it's called a dead space, okay? So that's the easiest way to explain it. And this will be important in the next slides with the VQ. Yeah, so what is a dead space? And there is two physiological components. Can someone tell me the two physiological components of the dead space?
Wait, Dennis, can I cut you off for a second?
Yeah.
This recording has been going for like almost half an hour when we were doing. I think. Stop it and then start it again or something, because
no, I started it now, the recording.
Okay, so sorry.
Okay. Yeah. So the two physiological components of dead space?
Anatomical dead space and functional dead space.
Yes, and you're right. The one is anatomical and the other one is the physiological. Basically, physiological is anatomical plus the physiological, yeah? The alveolar. So alveolar is basically all the alveoli that do not have, that do not participate in the exchange. Anatomical is everything that is not supposed to make an exchange, but the mechanical is not physiological, okay? So the mechanical dead space, like intubation and stuff, is not physiological. So we have the physiological is made up of the functional and the anatomical one. So we have the anatomical and the functional. These are the two components, okay? So that's the dead space, the physiological dead space. So dead space can be pathological, but can also be physiological. Now, for physiology, we talk about these two, anatomy and the functional, okay? So that's a dead space. So how much volume is a dead space, the physiological one? How can we increase and decrease it, and how do we measure this dead space? Do you know how to do it? How much is the volume of dead space?
The anatomical one is 150 milliliter, something like that.
I think it's a total. I think 150 is a total. Basically, 150 should be something like the total.
Like out of the tidal volume. So I remember that 150 is the anatomical and 350 is the like ventilating.
The ventilating. Yes. So 150 is the whole dead space. So how do we measure it? Is basically you take a guy, you make him blow in 100% O2. This is 100% O2. So it goes inside his lungs, and then he becomes like this. When he inhales it, he becomes full of O2. So his dead space is this. It's the O2, pure O2. Everything else in the lungs is going to be diluted with the nitrogen and all the other gases, carbon dioxide. But the dead space is going to be pure O2 because it does not participate in the exchange. And then what he does is he breathes out into a computer, And the computer, the sensor over here, will detect all the pure O2 first, because that's the one coming out first. And then it will detect all the diluted air, which was deep in his lungs. So this is the graph we get. First, he breathes in. So when he breathes in, the O2 is this part of the graph. And then he starts to blow out. So when he blows out, the first portion, which is O2, is going to be this portion over here. So this portion over here is basically the O2, which was his dead space. So this gray area is his dead space, all this gray area. So that's how you measure the dead space. Okay? Everyone's okay with this? How you measure the dead space?
Yes.
Okay, and then we have a way to increase it and a way to decrease it. To increase it, we can use some like snorkeling. You know what the snorkeling is? The one that people use to dive. So you actually make it longer, the anatomical one. You make the trachea like bigger because they have a longer anatomical dead space, let's say. And or there is some embolism, for example, less blood flow, because we said dead space is when there is ventilation, but there is no blood flow. So that's why embolism, we go back to the picture, embolism will count as dead space. Okay, but this is pathology. Yeah, so embolism, less blood flow. So these two things can actually increase the dead space, but this is pathology. This would be like kind of physiological, let's say. How do we decrease it? We can basically bypass, instead of having all this space here, that taking the dead space, we can just intubate into the trachea, and this is now shorter. So if you make an intubation into the trachea, there is less anatomical dead space. So these two ways you can actually increase and decrease the dead space. Everyone okay with this? We can continue to the next one. Okay. Shall I continue?
Yes.
Okay, so how does the dead space affect the gas exchange? We have positives and negatives. Can someone tell me some positives and negatives of why do we have the dead space? Like it can be useful, or the negatives that why we shouldn't have dead space. So why should we have dead space and why we shouldn't have dead space?
It makes lung more compliant.
It makes what?
It makes lung more compliant.
No, compliance is something else. We're going to talk about it after. So some positives would be there is filtering because of all the cilia that take out all the mucus and stuff. There is warming because it takes time before it reaches your lung, humidifying to change the composition of the gases that enter inside your lungs, as well as dephonation. So we can actually use our larynx and stuff to talk. So that's like useful things to have dead space. And negatives will be there is less volume available for gas exchange. As you can see here, this is the end of expiration. So at the end of expiration, the lungs are the minimum volume, and the dead space takes about 150 ml. Okay, so that's at the end of expiration. So what we do is when we inhale, the tidal volume will come inside the atmospheric air, 500 ml will come inside, and the dead space will actually migrate to the bottom of your lungs. 150 will go down. This will not be the fresh ones, and 150 will remain inside the dead space. So basically, the only fresh air you have is 350. Is everyone okay with this? Like how the dead space affects the breathing?
Yes.
Okay, so that's how we have less fresh air coming in. The dead space affects it. So you can see this diagram and you can follow it after. How when you exhale, the air comes off. You also have this dead space 150 ml, which is fresh, and the 350 which you just partially exchanged. Shunt versus dead space. We explained this. Okay, so which physiological process will compensate if a shunt happens? If you block the alveoli, do you know which process physiological process will try to compensate for this, for your ventilation. There is active and passive. So do you know any active process?
Like to breathe more?
Okay. So have you ever heard of hypoxic vasoconstriction? Hypoxic vasoconstriction. So basically this is a normal, the first one shows a normal perfusion ventilation. So the air is coming in is normal, the air is coming in is normal. You have the blood coming in, which is deoxygenated. Then you have the exchange happening, and then the blood goes out oxygenated. That's the normal, how it's supposed to be. However, when we have a blockade here, there is no air coming in, so there is no fresh air coming in here anymore. So when the blood comes in both ways, it's deoxygenated. Here there will be the gas exchange, so it will become oxygenated. However, here there is no gas exchange because there is no fresh air, so it is still deoxygenated. So we had a problem here, no ventilation, so this means it's called a dead space. No, it's called a shunt. So we have a shunt here. This is a shunt because there is blood, but there's no air. So for this shunt, what is going to happen is we are going to constrict the vessels here. We're going to make them smaller. You see, here it was bigger. Now it constricted. So the vessels detected hypoxia and they decided to constrict. Why do they constrict? Because the blood now will not go this way. It will all go to the good alveoli. So the good alveoli will actually have more gas exchange, more blood flow. So this is an active process and it's called hypoxic vasoconstriction. When there is hypoxia in the lungs, they will constrict. My question is, when there is hypoxia, will the vessels always constrict? Can someone answer this question?
What happens when the vessels, what happens in the systemic versus the pulmonary, which is, do you guys know, in systemic, when there is hypoxia in a tissue, what happens versus
Systemic
if there's
circulation dilate?
Sorry?
Yes, Aish is right. So they will dilate in the systemic circulation when there is hypoxia. And this is what we see. This is what we see in exercise. Remember with the physiology of exercise? And I showed you this drawing. So CO2 and everything will make vasodilation. So in the systemic circulation, there will be vasodilation. However, in the lungs, they will vasoconstrict. Okay, so this is the exception. The lungs are in the exception. Okay, only in the lungs. Everything else, they dilate. Okay, remember this point. Everyone okay with this? Any questions? Someone, please say yes or no.
So, so when we do exercise, somehow we can think it as hypoxia. Like in our body, the same thing happens. Like so in the lungs, it's a vasoconstriction, and in the systemic blood system, it's a vasodilation.
Exactly, yes.
Okay, okay.
So remember
Thank
that the lungs is the exception, okay? It's the opposite.
Okay, thank you.
This will be in one of your final exams to explain how the perfusion of the lungs is regulated. VQ ratio, do you know what is VQ ratio? What is V and what is Q? What does it mean VQ? V and Q.
It's the ventilation perfusion ratio, no?
Yes. So this is the ventilation and this is the perfusion. I don't know why they didn't call it VP, they call it VQ. So Q, remember, is the perfusion, okay? V is ventilation, Q is perfusion. How do they change from the top of the lung to the bottom of the lung? Do you know how V changes, the ventilation changes from the top of the lung to the bottom of the lung? If this is the bottom, the top, and this is the bottom.
It's on highest on top and lowest on the bottom, no? And
Opposite.
perfusion. Ah, okay.
So the highest V will be found on the bottom. Okay, so on the bottom you have the highest V. What about the Q? Where do you find the highest? On the top or the bottom?
Base.
What?
Base. Bottom.
Yes, base. Q is also found on the bottom. So basically the highest V and the highest Q is found on the bottom. Okay, so always remember that V and Q are higher on the bottom. Do you know how the VQ ratio change from top to bottom and why? And what is the average VQ ratio? Because I see that you know everything.
0.8.
Yes, that's the average, 0.8. And how does this change? Is it higher or lower on top and bottom?
At top is higher, at bottom it's lower.
Yes, so it changes like this. So basically, if you have the top, the red one is the perfusion, the Q, the blue one is the V, the ventilation. At the top, both of them are smaller, okay? They are smaller, both of them. However, as you go up to the bottom, down to the bottom, they become higher, okay? So both of them are higher, both of them are lower. However, what happens is the perfusion is increased, going to the bottom, is increased at a higher rate than the ventilation. So you can see the scheme here. The gray one is the ventilation, okay? So you see the alveoli, they are increasing as you go down the lung, they are increasing. However, the perfusion, which is this artery here, is increasing at a higher rate. So they both increase as they go down. However, the perfusion increases at a higher rate. And because this is increased, the perfusion is increased at a higher rate, it will make the ratio, which is this line over here, it will change the ratio. It will make it higher at the top and lower at the bottom. So the answer is the VQ ratio changes like this because the perfusion changes at a different rate than the ventilation. They both increase while going down the lung. However, because of the difference increase in ratio, different increase in ratio, they make this rate, different change of rate, they make this ratio difference. Okay, so in your exam, if you draw this graph and you draw the lung like this, like some 3 zones, and you explain how the perfusion is increased at a faster rate than the ventilation, then you can explain the VQ ratio. Okay. Any questions for this? Any question on this topic?
No.
Okay. So I said before we have passive and active. This is my handwriting from my second year. Yeah, passive would be something like recruitment. There is some recruitment maneuver. So some alveoli are not participating in the normal ventilation, but then after you do the maneuver for recruitment, they participate as well. So that's kind of a passive one. And the active is the hypoxic vasoconstriction. Okay. Yeah, so you can use this for your exam. The question was VQ ratio and homeostasis. Like how do you create the homeostasis on VQ ratio? You have passive and active. Active is the vasoconstriction and passive is the recruitment and distension. So yeah, that's for your final exam question. Next one. How do I move to make me breathe? Do I move the chest wall, the diaphragm? That moves the lungs. Do I move the lungs? That move the chest wall? Or C, I don't move anything and everything happens by themselves? A, B, or C? Who can answer?
C.
No, it's not C.
C is the best one. What are you talking about?
I know. Okay, it's not C. So of course the answer will be A. So I'm moving the chest wall, the diaphragm, and then this in turn causes the lungs to move, okay? So the next question is, the chest wall is connected to the lungs, right? So the chest wall is connected to the lungs. What is the structural connection between the chest wall and the lung? Which structure connects the chest and the lung? Do you know of any structural connection between the chest and the lung?
The pleura, no? But
Yeah.
they suck each other together, like it's because of the pressure.
If they ask you structural connection is nothing. There is no structural connection. It's because of the pleura. Okay. And the easiest way, okay, that's my next question, but you explained already that the pleura, how does the moving of the chest causes moving of the lung is the pleura. And the best way to explain it is what Kittner used in his lecture. So Kittner said, imagine you take microscopy slides. They come in the box. If you look at them in the box, they are touching each other. But it's so easy to just separate them and start using them. So the glass by themselves, they don't stick to each other. And they come in the package, touching each other. You can separate them. Everything is fine. However, when you do the specimen, you put 1 drop of fluid of water, the fixation, staining, whatever, and then you put the glass on it. And what happens now? Now you can slide left and right. You can actually slide this glass left and right. However, you cannot separate the glasses. So if you try to pull this glass up, you cannot separate it. So this is what Kittner used in his lecture, and I love this example. So you can say that 2 glasses together, they can separate. There is no problem. However, if you put 1 drop of fluid in between, they can slide. They can slide. You have to say that this is important. They can slide and they cannot separate. So they can go left, right, all directions. They can go in all the directions. However, they cannot go up. They cannot separate. So this is how the pleura works. So the pleura allows the 2 pleura to slide the pleural fluid. The pleural fluid allows the 2 pleura to slide on each other, left and right, up and down. So they slide, however, they cannot separate. And this is what Lisa said, that when you pull 1 glass surface apart, there is negative pressure and pulls both glasses back in. Everyone okay with this? You like this example with the glasses, microscopy slide?
Yeah, it's a good example.
This is Kittner's example. So we finish with the pleural. So the water drop in this example is actually the pleural fluid, and it's always negative. If you see, it's -4. Why is the intrapleural pressure always negative? You know why it's always negative? Why is not 0, for example? Why is not positive? Why it's always negative?
Because then there will be no like force to when you start breathing, so like push.
I agree with you.
know?
This is why we need it, but how is it achieved? Why is it always negative? What causes it to be negative? It has to do with the next 2 questions. Where does the chest wall want to go and where does the lung want to go? Do you know where the chest wall wants to go?
The chest wall wants to go outside and the
Yes.
lung wants to go inside.
Yes, correct. So these 2 pleura are always being tried to be separated. The chest wall will always try to push the pleura away and the lung will always try to collapse. So if you see the lecture, we have this joint. So this is the arrows. You see like the chest wall tries to go outside, the lung always tries to go inside. So the fluid is always under negative pressure. This is Pokorny's drawing. It's a bit confusing, but it's really nice drawing to explain it. So one line is the chest wall. This is the chest wall. And the other line is the lung. So this is the lung. So you have 2 lines. And then if you put them together, you get this thick line, which is called the combined lung and chest. It's here, lung and chest combined, okay? So it looks a bit confusing, this graph, but if you can draw this in your exam and explain why it's negative, if they ask you, it's really helpful. So there is the volume of the lungs, breathing in and breathing out, and the pressure of created by the chest wall and the pressure created by the lungs. And if you put them together, you get this line. So basically, when you have the resting chest volume, it means the chest wall is not recoiling neither in, neither out. So it doesn't want to go anywhere. It's, wait. Should be here. So this point over here creates 0 pressure because the wall doesn't want to go neither in, neither out. So this point over here is where is the volume that the chest wall is perfectly fine with it. This point over here, is the point where the lungs do not want to go either in, either out, and it is actually the minimum lung volume, is completely collapsed lungs. So when you have completely collapsed lungs, for example, in pneumothorax, you have zero pressure. So the zero pressure is where the lungs doesn't want to go anywhere and the chest doesn't want to go anywhere. And this is, if you put them together, they create this point over here, which is the functional reserve capacity, where the lungs and the chest don't want to go anywhere. And this is actually the end of the quiet expiration. It means the lungs collapse equals the chest wall expand. Okay. Oh my God, it's more confusing than I thought. Do you want me to explain it more or just skip it?
Guys, tell them if you wanted to be
I
explained
want to
again.
know where is in the previous slide there was the 756 millimeter. Where is it from?
This is the absolute pressure. However, when we talk about the lungs, we say relative pressure. So you have absolute pressure if you actually measure the pressure, and the relative. Relative to what? This is relative to the atmospheric pressure. So if we say atmospheric pressure is 0, then this would be -4. And this would be, for example, 0 or something, and -4. These are not the actual pressures. If I take my invasive catheter pressure meter and put it inside, it will be, okay, this will be -4.
But 1 atmosphere is 760, no?
760, yes.
No, so how do we know it's -4 and not -5?
So it would be, sorry, it will not be absolute. It will not be absolute -4. It would be absolute 756. So relative to the atmosphere, it would be -4.
Yes, so what he's trying to say is if it depends upon the instrument. If you configure the instrument to measure the atmospheric, if you configure the instrument so that the instrument thinks that the atmospheric pressure is 0, then it would calculate a -4 and 4 millimeter per mercury. But
No,
if you
no
configure,
problem. But why is it 4 and not a different number?
why it's
It's just from the book. It's like resting.
Yeah, that's what keeps the lungs open. Otherwise, if it was any other value, they would probably collapse.
It's 4 at this point and at this point. However, when you breathe in and breathe out, so you see that this is breathing in. This is just breathing in, okay? No. See, this is inspiration and expiration. So inspiration, expiration, you have change of the transpulmonary pressure. It becomes bigger and bigger. And at the end of inspiration is actually the biggest, okay? So it's like -7.
So do you get why? So the reason is if it was anything other than -4, suppose if it was -3, so there's a certain amount of volume of the lung that is necessary to keep the lungs open here. And that volume will come if the resting pressure is -4. And if like in case of pneumothorax, when the pleural pressure becomes positive, so when it becomes something other than -4, suppose positive, that's when the lungs collapse. Does that answer your question?
I'm not sure, but
What is the question again?
no, just like I didn't understand. No, like I understand the point and why and blah blah blah. But like,
Yeah.
if because like I know Vlad and he's gonna ask me why is it -4 and not -10. So what do I say to him?
So what you have to say, I think, is -4. So based on the lung compliance and the chest compliance and the lung compliance, am I right? Salius, based on those two, this is the value. And
this is basically the value for a 70 kilogram 20-year-old male, which is healthy.
So it can like vary a little between people?
Yes, and also during inspiration and expiration. As you can see, end expiration is 4, like for our case, which is man, 20-year-old, 70 kilogram, it will be -4. and the resting conditions and everything. However, in end inspiration is bigger.
Because the lungs want to go back to normal shape.
Yes, and the chest wall also wants to go back to normal shape, which is out.
No, normal shape is
Out.
like
The chest wall.
The
normal
chest
shape,
wants to expand. The lung wants to collapse.
and then normal lung is here. So the lung is pushing the wall back to this position, and the chest wall is pushing back the lung to this position. So they pull on each other. So when you have pneumothorax, the chest wall will actually be bigger than normal.
Yeah, and pneumothorax is the best example, yeah, because in pneumothorax, the chest wall becomes the ideal position where it wants to be, and the lung becomes to the position where it wants to be when there is no pressure involved. But when there is not pneumothorax, that is when the pleural membrane connects both the lungs and the chest wall, they meet at a certain equilibrium point where it's not completely expanding the chest wall and not completely collapsing of the lung.
okay, thank you.
No problem.
Any other questions? Any other questions on this transpulmonary pressure?
No.
So I'll send you the presentation and you can read it after, like you can see the images and everything. I want someone to explain me the inspiration using the following terms. So the initial status, the inspiration, and the final status. Using these terms, it's easy. I even put them in order. Can someone try? Let's see. Ace? No. Kirari?
A moment, I still need time to
Okay.
yeah summarize these words and.
Okay, I will try to explain these words, what they mean. Intrapulmonary pressure is basically the pressure found within the lung. If it's 0, it means it's actually the same as atmospheric pressure. If it's the same as atmospheric pressure, there will be no flow, because we know that the air and all the fluids, they go from high pressure to lower pressure. So if both of them are 0 means atmospheric pressure. So if both of them are the same, there is no flow. So we have the intrapulmonary pressure, airflow. So you can start by saying this as the initial step. There is no airflow because the intrapulmonary pressure is the same as the atmospheric pressure. Breathing muscles, I think you know this from anatomy. Intrapleural pressure is what we just explained, which is the pressure within the two pleurals, the parietal and visceral. Transpulmonary pressure, it means what is the difference between the pleural fluid and the lung. So this is a transpulmonary pressure. So, okay. Then we have the lungs. You know the lungs. Intrapulmonary pressure is the same as the first one. Airflow is the same as the first one. You just need to explain what changed. And again, it will continue. This process will continue until what happens? Until when? So I think it's pretty straightforward. Does anyone want to try to explain this? Because if you can write this down, it's basically physiology of breathing.
You guys can try. No, don't worry about getting it wrong. It's okay to get it wrong.
So the first 2 times, we can say that when the atmospheric pressure and the intrapulmonary pressures are equals,
yes,
airflow doesn't happen.
Yes, good. Then what happens? So you're done with this. Good. Then what happens to inspire?
Then breathing muscles such as diaphragm and also skull and muscles, like these muscles, they pull the chest walls.
They
somehow.
contract, yes.
Yes, and then it causes intrapleural pressure goes to minus.
Even more negative.
Okay.
Right?
Okay.
So you see here, it's supposed to be -4. It became -5 or -6, you know, like it increases even more negative. Yes.
And then transpulmonary pressure. What happens for the transpulmonary pressure?
So
it goes to negative.
Yes, and pulls the lungs.
Okay.
It pulls the lungs towards where?
To outside.
Yes.
And then intrapulmonary pressures goes to more negative.
Yes.
And the airflow happens.
Yes. So airflow goes inside the lungs or outside?
Inside.
Inside the lungs. Good.
Yes.
And how until when?
Until intrapulmonary pressure goes to more positive.
No.
Go back to the previous state.
Which is what?
Atmospheric pressure.
So 0, yes, atmospheric
Zero.
pressure, which is 0.
Yes.
And then the airflow, what happens to airflow?
Airflow, it go outside.
Yeah, no, airflow goes back to 0. It stays there.
Okay.
This is the end of inspiration. So you explain this process from here until here. Okay?
Okay.
So the air tries to go inside and then it stops going inside. That's where it stops. Okay? So this is inspiration.
Okay, thank you.
Good job, Kerari.
Good job, Kerari. Then we have the expiration. It's basically the same but opposite. So you start again with the same. Both of them are the same. There is a 0 pressure, airflow is 0. And we know that we will end with 0 and 0 as well. So what happens in expiration? The breathing muscles relax. The intrapleural pressure is still negative, but less negative. So if we go back to our diagram, it's still negative, but starts to become less negative. Okay. If you try to do force expiration, it can even go to positive. If you use all your abdominal muscles to contract and blow forcefully, it can even go to positive, like blow inside a balloon or something. The lungs would collapse, of course. Yeah, and the air is going out. So that's the expiration. Pneumothorax. What happens to the intrapleural pressure in pneumothorax? What happens to the chest wall and the lungs? 3 things. Who wants to try? Katie?
No, I don't want to try. I want to listen.
Okay, Lisa, you can say.
Okay, so the intrapleural pressure will be 0.
Yes.
The chest wall we want to be at its ideal position,
Which
so
is?
expanded.
Expanded, yeah, outside.
And the lungs also we want to be at their ideal position, so they will collapse.
Collapsed. So it's basically it's not. I didn't put a photo. Okay, so it's the lungs will collapse, and chest wall expands, and this will not be negative anymore. So that's a pneumothorax. I should have put a photo here. Everyone okay with pneumothorax? Remember that the chest wall expands. It will go even bigger. So when the patient comes, you can look at him. He's going to be suffering, but he's going to have a huge chest wall. And you're going to be like, hey, his lungs are fine. Yeah, but if you look inside his lungs, they're so small because they collapsed. So that's kind of a pneumothorax. Okay? What is lung compliance? I want someone to tell me what is lung compliance. Who said about lung compliance before? Was it Ace? Ace? Are you still with us? Ace? Okay, we lost Ace.
Maybe someone else.
Maybe someone else?
Isn't it the extensibility of the tissue? Like how much you can extend?
Okay.
Delta volume over delta pressure. I just remember that. I forgot the definition. It's like distensibility, something like that.
So distensibility, yes, it's not just the tissue, it's also the fluid. So basically, it's how much the lung volume can expand based on the increase of transpulmonary pressure. So basically, how much volume the lung change based on how much difference of pressure. So it's the distensibility of the lung during inspiration. So you have to you're trying to put the air inside the lungs. Okay, so it is a change in volume for a change in pressure. If I need a lot of pressure to make the same volume, we call that, okay, the lung is not compliant. I'm trying to put the air inside, to put the pressure inside, but it is not expanding. If the opposite happens, if I get so much volume increase with just a slight pressure change, then we say the lung is compliant. Okay? So if it's harder to put air inside, it's less compliant. Okay? So you look at the kid blowing the balloon. You know that if you try to put in a hard balloon air and you try a lot, it doesn't expand. This is less compliant. But if I just breathe really fast inside and it's easy, we can say the balloon is more compliant. So,
you know, actually, that's a good question. I always confuse the definitions and explanations of compliant and elasticity.
So elasticity has to do only with tissue, but compliance has to do with change of volume per change of pressure specifically, which also includes the fluids inside. It's not just the tissue. I will show you after. I have a drawing from my second year. So by what I just said, do you know any factors that can make it harder, less compliant, the lung to expand?
If it has more fibers.
Yes, more collagen fibers, less elasticity. This is called a restrictive disease, okay? Restrictive disease. For example, fibrosis. Fibrosis means a lot of collagen. There is no elasticity. It's like a rope. It's not like a balloon anymore. So it becomes hard, stiff lung. So this is less compliance, okay? So that's a restrictive disease. Do you know Laplace law? Can someone say about the Laplace law? It's physics. You know Laplace law? Anyone? Okay, if you don't know Laplace law, then just answer the next question. If I have a small balloon and I attach them to a big balloon and I have this in the middle so the air does not move anymore, and then I decide to remove the valve, is the air going to go from the big to the small or from the small to the big? So this is the experiment in life. You have a valve which is locked. You put a small balloon and a big balloon. And then you say, okay, now I'm going to do the experiment. You remove your hand, and then does the air go from the big to the small or from the small to the big? That's my question.
I think it goes from the small to the big.
Yes.
Because like the small is more resistant to change in shape, if that makes sense.
Basically, the answer is Laplace law. If you say Laplace law, you're covered. So Laplace law says that if you're smaller, you have more pressure inside. You're pushing harder. If you're bigger, you're not pushing in as much. So if I connect them, the air will go this way because he's pushing harder. And it will become even smaller, and it will push even harder and harder until there is no balloon left. All the air went into the big balloon. So this is the experiment to show the Laplace law, okay? So the same thing with the alveoli. If I have one alveoli which is small and one alveoli which is big, where do you think the air will go in this situation? From the small to the big or from the big to the small?
Small to big then.
Yes, and what will happen is that this will collapse and this will explode, okay? So is there all our alveoli in the same size in our body to avoid this? Or this actually happens in our lungs? We have some alveoli bigger than others.
We said that they change shape as you go down the lung. They become bigger.
Yes. So we do have some aniso size. Okay, so they're not always equal. And how do we solve this problem?
Surfactant.
Yes. Oh my God, you know everything. Okay, yeah, basically surfactant. Okay, how to solve the problem is surfactant. So if we don't have surfactant, then it will go from the small to the big. This will collapse. This will explode if there's too much air. So if we put surfactant, then this will change the, let's say, the recoiling of the lung. So two things about okay, what is the role of the surfactant? What cells produce it? And what's the problem of the premature babies?
The surfactant is to reduce the surface tension.
Yes. Which cells?
The like how they called the second one, the second type one, the
yes.
alveolocytes, the type 2, and because they produce surfactant after like 27th week or 28th week or something like that, they don't have it yet.
So what
like
happens
the
to babies?
Their lungs will collapse, like the alveoli will collapse.
Yes, correct. This is what happens. So compliance is made of what? We have the tissue, all this elasticity, and also the fluid, okay? So remember, explaining your exam, you have a question called lung compliance, elastance, and surface tension. So you can say compliance is made of the tissue elasticity, of the fluid, explain the droplet experiment. You can say we have the surfactant to solve this problem. You can say there is a Laplace law, and there is premature babies. You can go even further explaining what is the composition functions of surfactant, whatever. And then you can draw this drawing, the same drawing, explaining what happened just with water and what happens with surfactant. How do we solve this problem? Like the air goes there. So that's how you answer your final exam question done, 87 question done. So compliance is made of two things, okay? It's not just the tissue, it's not just the fluid, and they make difference of volume per difference of pressure, okay? How much volume change happens over how much change of pressure happens. So that's done with compliance, okay? So you know this as well. Work of breathing has to do with the compliance, surface tension, the airway resistance. There is also a question called work of breathing. That's why I explained lung compliance and the surface tension. Airway resistance, you should know already from anatomy. It has to do with the diameter and stuff. So this is another final exam question done. Regulation of breathing. So do you know regulation of breathing? It's one of the hardest topics. You have the peripheral chemoreceptors, so central carotid body, etc. You have to know the mechanism like potassium, depolarization, and then you can just say the nerves, glossopharyngeal and vagus nerve, how they go, and they go in the medulla. And you say in the medulla we have the pneumotactic center, the dorsal, the ventral group, how they come. We said vagus and glossopharyngeal, okay, and then they will have the processing here, and they will have the efferent respiratory motor pathways, which is the phrenic nerve going to the diaphragm. So that's the peripheral. And then in the central, this is a bit more complicated. It has to do with CO2. It goes in the CNS, the CO2 becomes acid, and then the acid will say how much CO2 it has to the brain. So that's the chemicals, the nerves, and another thing to remember about the brainstem is that we also have a bit of voluntary control. So when we speak or when we want to hold our breath, to go underwater, etc. So also we have voluntary. And then we have some wave shaping. This is a bit more complicated. This is Pokorny lecture, and I added some extra things on the bottom. Yeah, it's how you make the rhythm and the waves. It's a final exam question, wave shaping, I think. And the reflex. We have the reflex of mechanoreceptors in the alveoli, the stress receptors, the conscious control we talked about. So this is how you regulate the breathing. Panshu, do you want to say anything about this?
No, I don't have anything. You said everything.
So these are important for our experiment. Like how can we actually change our breathing? Okay, and that's it. I'm done. I'm done, me. Any questions about mine before I stop sharing? Any questions?
If you guys have any questions, ask now. So before that, so I can continue. No questions?
No.
Okay. Okay, so for what I'm going to be doing is I'm going to be discussing the practical part. I hope you guys were able to do the practical part. Yeah. Do you guys remember anything from the practical part of spirometry?
Being close.
Sorry.
Being close.
Being close. Okay. I didn't get what Ace. Ace, did you say that?
Lost.
Lost. Lost. Okay. Sorry. So no worries. I will. I mean, if you guys remember the do you guys remember what you guys did in the lab? So So I'll give you a basic review. So we had two instruments, right? We had the spirometer and we had the PEF meter. These are the two instruments we used. And in the so the main goal of the PEF, so PEF meter is the peak expiratory flow meter, and the spirometer we will discuss. So the main aim of the lab was to figure out if a person has a normal, restrictive, or an obstructive disease, right? Now the PEF meter will what we will discuss more on it, but what the basic idea was that we will use the PEF meter to detect any kind of disease. So it can be either obstructive or restrictive. And then once we detect that it's the person has abnormal values, we will go to spirometer to detect if it's restrictive or obstructive. This is the main idea. Now we have to so the way this presentation is divided, that I will first discuss about the spirometer, and in that first we will discuss about the terminologies that you will require to understand. So the thought process should be, first we will understand the values and definitions of certain topics, and then we will integrate those definitions into graphs. And then once we get the graphs, understand how the graphs work, and we will use those graphs to differentiate between obstructive, restrictive, and normal. Does everyone get the basic idea of how I'm going to discuss all of this? Because it's important to have a thought process, otherwise you will get lost again. If you want me to just give a thought process again, I can say it again. Or if you understand, you can tell me you understand. Anything? Guys, just say yes or no, at least I'll know I can continue. Hello.
Yes.
Yes. Okay. Finally. Okay. So let's go to the first one, which is a spirometer. So if you guys remember, the way we did it was that first, this is the kind of machine we had in our lab. We take the spirometer, and what we do is we breathe in as fast as possible through the spirometer and then breathe out. And what we get on the screen was something like this, or this is the graph. And so the way so the way this value was obtained is basically the top part is the expiration. This is expiration. Let me put this. Sorry, 1 second, guys. Yes. So the this top graph is represents the expiration, and the graph below represents the inspiration, right? So if I were to modulate this, how it would look like is I breathe in and then I breathe out as fast as possible. So I breathe in, I breathe out as fast as possible for how much ever time I can, and then slowly I'm unable to breathe as fast, so slowly I breathe out slower and slower, and slowly come to the point again, so to a point where I can't breathe out anymore. So this is what we got, and I'll discuss more on it later. So we'll go first to the static. We'll first understand the terms that we will later on use. So first, we have to understand what are static indicators. Does anyone have any idea what these static indicators are, and any small definition? Anyone? Okay. Okay, so static indicators are the values, so volumes and capacities that remain static in time, so they don't change with time, versus the dynamic indicators. Those are the ones that will change with time. Static indicators includes the tidal volume, the expiratory reserve volume, the inspiratory reserve volume, the residual volume. So can anyone tell me what is tidal volume? Any idea? A small definition will do. Anyone? Just try it, it's okay.
Like the amount of or volume of air that you breathe if like you are relaxed, let's say.
Exactly. So the volume of air that you breathe in and out in a normal state. Exactly, that's correct. What about expiratory reserve capacity? Any idea? Any idea, guys?
The volume that expired after the expiration of the tidal volume.
Exactly. It's the forced, if you were to force the expiration, this is the amount of volume we can expire. And same goes for inspiration. If you were to inspire forcibly, this is the amount of volume we would inspire plus the tidal volume. So, and can anyone tell me what is the maximum tidal volume that we can have? So we know, I think everyone knows 500 ml is the normal one. How much can we have? How much can the maximum tidal volume be? This is something
as much as vital capacity.
Exactly. This is something Professor Vlad will ask you if you ever get any question related to respiratory system. So exactly. So you got it right. Tidal volume, maximum tidal volume is equal to the vital capacity. So and residual volume, I think everyone knows it's the volume that that resides in the lung after forcible expiration. And with these values, we can calculate capacities. Yeah, like you said, there's vital capacity, there's functional reserve capacity, there is the inspiratory capacity and total lung volume. Anyone? So can anyone quickly give me any approximate values on these? Any range, anything will work. Just try recalling some of the values. Let's start with this one. ERV. Any guesses? No?
1100.
Yep, exactly. Exactly. What about IR, inspiratory reserve volume?
3000.
Mm-hmm. What about residual volume?
I don't know.
Sorry? Anyone?
Like between 1,000 and 1,500 or something.
Yep, exactly. That's a good range. 1,000 to 1,500. Okay, so what about vital capacity? We just add the values we just mentioned.
Around 5,000 ml.
Yeah, exactly. And what about the total lung capacity? What can, if vital capacity is around 5 liters, how much can we expect the total lung capacity to be?
Around 6.
Yep, 6, 7, no problem. Okay, so we discussed the static indicators. This is the graph. I think, do you guys know this graph? I think you should know this graph. You don't want me to explain this graph, right? If you do not know the graph, then I'll have to explain it.
No, continue.
Okay, perfect. So what are dynamic indicators? So we discussed the static indicators, we know what they are. Now we have to discuss what are the dynamic indicators. And for there are many actually, and I will only discuss two important ones because these are the two we'll need to understand the differences, and we'll need for understanding the graphs. So any idea which two I'm talking about? I know it's a wild guess, but if you have studied this, you might have an idea. Okay, just tell me what are dynamic indicators. Okay, so what dynamic indicators are is the values, capacities, and volumes that change over time. So again, like I said, there are many of them, like the respiratory rate, there's the minute ventilation, there's the minute alveolar ventilation. But the two important ones we require are these two. We require forced expiratory volume in 1 second, and we require FVC, forced vital capacity. The reason we require these two is to get this ratio, because this ratio is what we will again later on have to use to differentiate between obstructive and destructive and normal. There is another dynamic indicator called maximum minute ventilation. Does anyone have, can anyone give me a quick idea of what it is? Any idea?
Guys, this is a laboratory you did already, so at least
yeah, this is something. It's okay if you guys don't remember a lot of stuff. I can understand that. But if you guys just try to recall something, because if you recall now, it'll probably end up helping you in the final exam.
At least try to remember what you did.
Okay, so what maximum? Yeah,
sorry.
go ahead. No, please go ahead.
Yeah, I'm just trying to remind myself. Minute ventilation, like
Right.
the normal value, it's like 5 liter or something, if I remember properly.
Minute ventilation is.
I don't remember.
Yeah, 1 second. Minute ventilation is tidal volume into a respiratory rate. So around, yes, 5 liters. Yeah, if the respiratory rate was 10 and then 500 ml is what we consider the tidal volume, 5 liters would be the normal minute ventilation. Exactly.
So maximum minute ventilation, it can be 20 times
vital capacity, approximately vital capacity into the maximum respiratory rate. But again, I don't want you to tell me the value. You're actually right. You have the right thought process in it. But what about the definition? Is the maximum amount of air that maximum amount of volume or rate? Is it volume or rate? That's the question. I think this is the main idea behind asking this.
It's volume?
Yes, exactly. So it's the maximum volume that a person can breathe in and out. Why is it dangerous? If you ask a person to do maximum breathe in as many times as possible and as much air as possible, why can it be dangerous for him or her? Think about it. You're breathing very fast. You are taking out. You are breathing very fast. So that means what can happen? You're losing a lot of carbon dioxide very quickly. What is it called? Any idea, guys?
Because you can faint.
Exactly. Yeah, exactly. Hypocapnia. So because you breathe too fast, you're gonna get hypocapnia. And hypocapnia is basically low levels of CO2 in the blood, and which is why you can faint. That's exactly right. Okay. So 1 second. So we understand the two kind of indicators. Do you guys understand the FE1, FEEV1, and forced vital capacity? So forced expiratory volume is the amount of volume that you would exhale in 1 second. Forcefully exhale in 1 second, and forced vital capacity is actually just the vital capacity again. It's just that it's the vital capacity measured when there was a forceful forcible conditions. I mean, it's again the values of vital capacity and forced vital capacity would be the same. So we understand what is FE, the forced vital capacity. We understand what is the forced expiratory volume in 1 second, and we can use that to calculate this ratio, which again we will later on use to differentiate between the obstructive and restrictive, and again normal. Okay.
So basically, when you say to someone, breathe in a lot of air, like your maximum air, and now I want you to breathe it out as fast as possible, the air is going to come out. However, 80% of this air should be within 1 second.
Exactly. Do you guys understand?
Yes.
Okay, perfect. So now we understand the static indicators. We understand what the dynamic indicators are. We also understand the two main one which we'll be using this time in this presentation. And now we will use all of this to understand these two graphs. Okay, whatever we learned during these two top topics, we will use that to integrate it with these two graphs. And then in final, after we understand the graphs, we will connect it to these diseases. Okay, so we have two graphs. We have the flow volume, we have the volume time. Flow volume is what you would get in the spirometer. This is the result you would get. Volume time, it's the graph that again, so you in this one, it represents the expiration and the inspiration. But in this one, this is again the same result, but in a different form. So it's volume time. In this, we don't need the inspiration, we take the expiration. So if I were to breathe in, hold, and then breathe out, it would come like this. So if I were to breathe in, breathe out as fast as possible, and to the completely, this is the graph I would get. And it's again volume versus time. So I start breathing at start breathing out at 0, and I breathe out as much as possible in the first second, which is approximately 80%, like Stelio said. And then after that, I breathe out the remaining volume of air. So these are the two graphs. Do you guys understand how the graphs work? Okay, I'm going to move on. Okay, so you need to understand the two graphs. And now we understand the graphs. I'm going to ask you a quick question. Based on this graph, can you tell me these four values? So approximate values from this. So we know this is flow and this is volume. The x-axis is volume, the y-axis is flow. Based on this x-axis values, can you tell me what is the residual volume and what is vital capacity, total lung capacity, and which point of this graph would be the PEF, the peak expiratory flow? Let's start with the residual volume. Any idea how much of it, what could be the value? So we have 1 liter, 2 liter, 3, 4, and 5 liters on the x-axis. Any idea what would be the residual volume based on this graph?
3.8.
Why do you think it's 3.8?
Because the residual volume is at the end of the expiration, no?
It's the volume that is left out, right?
Yes.
Exactly. So if let's say I start expirating all the way through, and this is the amount of air I have expired, right? So 1 to 3.8, so let's say, is the amount of air I have expired out. So 3.8 liters would be the air I just expired out. The volume that is left inside the lung would be the volume after. So it would be this volume, right? So that volume would be the residual volume. Do you guys get it?
So we cannot measure it.
1 second. Exactly. So this is just to understand the graph, right? Actually, you're right. You cannot measure because as long as you don't know the total lung capacity, you cannot measure it. But for the understanding of this graph, and to understand how how we actually differentiate between the three restrictive and obstructive, we need to understand how that static values are connected to this graph. But you get it after the flow, after I take out the 3.8 liters of air, if I were to say the lung capacity was 5 liters, the residual volume would be around 1.2. Yeah.
However, we need to know the total lung capacity.
Exactly.
If we don't know it, we have to use helium. So it's another test. It's not this one.
Exactly. So if for this, I should have actually said, for the sake of examples, let's say the total lung capacity is 5 liters. Yeah. Based on this, we calculate the residual volume, which is this volume. And what about the vital capacity? Any idea? vital capacity would be the total amount of volume you would breathe in, total amount of volume of air that you would breathe in and out forcibly. So I just told you about the graph saying that I breathe so much air out, right? What 3.8 liters would be the vital capacity. Again, total lung capacity is 5 liters. We figured out 1.2 liters is the residual volume. So the vital capacity, total lung capacity is equal to residual volume plus vital capacity, right? So based on that, you can also calculate the vital capacity. Now, can anyone tell me where on the graph is the peak expiratory flow? So we have the flow on the y-axis. Where is the peak for expiration? And what is the value? Any idea? You can see on the graph. 7, exactly. Because during the expiration, this point is the peak peak point in the flow. This is where we would calculate the peak expiratory flow. So we have connected some of the static values to this graph to understand how this graph functions. Now we're gonna so again, this is the same thing. Now we're gonna see the normal. We're gonna compare it to the obstructive, and then we're gonna compare it to the restrictive. In the normal, again, we have said that this is how the normal graph looks. We have about 6 liters vital capacity. Let's say around 1 liter residual volume. So vital capacity, residual volume, and then total lung capacity. This is what we understood in the flow volume. And in the volume time, we have discussed that the way this graph is formed is that if I were to expel all the air out as fast as possible, in the first second, the amount of air I expel out is the forced expiratory volume in 1 second, and then I keep expelling the air out as long as I possibly can, and then this whole volume would be the forced vital capacity or vital capacity. Do you guys understand until now what I just said? Because if you, I will have to explain it again, otherwise you won't understand the next sections of this presentation.
I have to make a note that these are used to find the dynamic values because we don't know the total lung capacity. So theoretically, if we know it, yes, we can use all these graphs to find all the static ones. However, we use the other graph, the static one, which is the x-axis is time and the y-axis is the volume, to find all these capacities and volumes.
This one, right? This one.
Yes, this graph we use for to find all the static indicators.
Exactly.
So the other one we use to find dynamic. However, no one knows the total lung capacity. It's not easy to do.
Exactly. So so that's the thing. In order for us to understand it, we have to connect these two flow volume to the static values. I know it's confusing because I was confused for the past 2 days because I was like, why is it connected? But unfortunately, every book that explains this relation with the restrictive and obstructive and normal, they're always correlated to the residual volume, vital capacity of the lung in the flow volume curve, and that's why I have to explain it. I know it might not be clear, but if you have any doubts, just ask now. I'll try to clear it as much as possible. Any questions, guys? Shall I continue? I'm going to continue, okay? Okay, so first one is obstructive. We know, so we know that in obstructive there's emphysema and there's bronchitis and asthma. These are the two diseases that are categorized under obstructive. The way they work is that let's pick up emphysema. In this, what happens is there's a loss of so emphysema is when there is loss of the elasticity of the lung. So due to in the pathological process, I'm not going to discuss what happens due to the pathological process of the emphysema is that the lung loses its ability to collapse back, which is the loss of lung elasticity. And it's also because it cannot collapse back, if you breathe in, you are unable to expel all the air out. That means the lung expanded stays expanded for a longer period of time, which is gain of compliance. It will have a bigger compliance. So the lung can expand, but it cannot compress completely. So what would happen is that because if you suppose breathe in, you cannot breathe all the air out. What would happen is there will be air remaining in the lungs. There'll be air that will still remain in the lungs. That means there'll be increase in the total lung capacity. There'll be increase in the there'll be increase in the residual volume. There'll be increase in the expiratory reserve volume. But because let's say you breathe in, you cannot breathe all the air out. But the next time you breathe in, you cannot breathe in the same amount of air. You have to breathe in less because you already have your lungs filled with the previous load of air that you couldn't breathe out. So there'll be a decrease in the inspiratory reserve volume. But in total, there'll be increase in the total lung capacity. There'll be increase in the functional residual capacity, and there'll be approximately the functional vital capacity would remain approximately same. Now, based on these three things, we construct the graph. So let's come to the first flow volume curve. Yeah. So to construct this graph, to construct the graph for obstructive, we know that the residual volume increases, right? Now let's take that residual volume. This is the normal, right? And this is 0. This is 6. Residual volume increases, right? And this is the new residual volume. So residual volume has increased relatively. And we have also said that the total lung capacity also increases. Now again, if the total lung capacity increases, the graph actually, so instead of total lung capacity being this, it increases to this. There's a small increase over here. Do you see that? So do you guys understand why? So because there is a shift of the, let me just remove all the, can't remove it. So we have discussed that there's the based on the previous understanding of how the disease works, we have constructed a graph based on the previous understanding. So there's an increase in residual volume. The graph shifts to the left. There's increase in the total lung capacity. So again, the graph shifts to the left. This is why there's a left shift. But now we have to understand why is there depression in the peak expiratory flow. Does anyone have any idea why the peak expiratory flow decreases in obstructive diseases? Any idea?
The alveoli collapse during expiration.
Yes, exactly. So, but the peak expire. Well, so the PEF depends upon the fact that how fast or how much volume per unit time you can exhale, right? So if I were to, but for what happens in obstructive is that let's take the example of bronchitis and asthma now. What happens is that there is narrowing of the lumen. When there is narrowing of the lumen, there is less amount of volume that can pass through the lumen per unit time. So because of which the ability of the person to forcefully exhale the air out is less. Because for the first example, emphysema, because the lungs, exactly like you said, because the lung lose elasticity, they are unable to collapse back. So let's say you breathe in, the lungs are open, but because of the lack of elasticity, they don't collapse back together to the original position, because of which the amount of, again, the volume flowing through the trachea per unit time is lower, and that's why the PEF will be lower. Do you guys understand?
Yes.
Perfect. Okay, so we understood how we construct the graph. We have a normal graph, and in obstructive, we understand that the two things go up. just remember two things. If nothing goes, if just to be able to construct the graph, the total lung capacity goes up, the residual volume goes up to construct, and then we take the graph, we shift it to the left because both of them are going up. And the second thing we do is we drop the PF because, again, like I explained, just remember this thing and you'll be able to make this graph. The kink over here represents the obstructive, represents that there's an obstruction in the trachea. Now for the second graph. So the second graph, again, this is the normal graph, right? This is what we have discussed now. What happens, again, we have to go back. So we have said that the vital capacity will remain approximately same, but the forced expiratory volume, again, we can refer to the PF, that it's going down. So the forced expiratory volume in 1 second, that means the amount of air I can just forcefully send out in 1 second is drastically lower, because again, my lung cannot collapse. In emphysema, in bronchitis and asthma, there is inflammation in the trachea. So again, there's the lumen is low, the lumen diameter is decreased. Both of them result in the FEV going down. So instead of me having the FEV at 3 liters, it becomes 1 liter. But because we have discussed the vital capacity remains same, this is how the graph is formed. So we use these two values, FVC and FVEV1, these two values to construct the graph, and then we can use the ratio. And the ratio, suppose if I take 2 liters and 5 liters, so the FEV1 is 2 liters, forced vital capacity is 5 liters. The ratio would be 0.4, right? And that's less than 70%. Do you guys get it? Do you guys get how the graphs are formed and how they connect to the values we discussed and how these values come connected to the diseases?
Yeah, got it.
Okay, perfect. So we understand how obstructive works. We understood the thought process of how that diseases causes some physical change in the lungs. This physical changes result in the changing in the volumes, and how these volumes then change the graphs of the two graphs we are talking about. Okay, for restrictive, now let's take fibrosis. There's fibrosis, asbestosis, and sarcoidosis. Let's take the example of fibrosis. In fibrosis, what happens is, again, like I think Stelios mentioned, that because so the lung tissue is replaced by a fibrous tissue. The fibrous tissue is stiff and hard, so the lungs become hard. That means so the lungs become hard and they're unable to expand a lot. If they cannot expand to their original volume, the total lung capacity. So let's go back to the basic ones. The residual volume goes down, all the volume goes down. So residual volume, expiratory reserve volume, and inspiratory reserve volume. All of the volumes go down. As a result, the total lung capacity goes down. The vital capacity, forced vital capacity goes down, and so does this. Now, so total lung capacity is going down, the vital capacity is going down, the residual volume is going down, everything is going down in this restrictive. Based on that, we will construct the graph now. Now again, we have the residual volume. So residual volume is moving this way because it's decreasing. The total lung capacity is moving this way because it's decreasing. But in this case, we don't see the huge drop between the peak expiratory flow. Any idea why? Why the PEF doesn't drop like it did in the previous one?
Because the elasticity is increased.
Elasticity, well, you have the right thought process, but not the elasticity, because it's fibrosis. Well, okay, yes. If the elasticity has increased, the compliance would decrease. No, wait, compliance would decrease. But I'm asking, what I'm asking is why the PEF won't change approximately. It's approximately the same. in this case? Any idea why?
Because
Because- Oh, sorry. Go ahead.
No, you continue.
Anyone try? Both of you can try. One of you can go first.
Okay, so I start first.
Okay.
So in this case, the airflow, it's not disturbed. So
Exactly.
yeah,
Exactly.
the speed, it doesn't change.
Exactly, because yes, exactly, because in the restrictive case, the only the problem of the lung is not that the lung cannot push the air out, because it has so much fibrosis that the issue is not with the air being expired out. Instead, it's the problem with the air even coming in. Because okay, let's go back actually. So the problem with restrictive is air to come in. Just remember this mnemonic to remember. Remember what restrictive does. The problem with obstructive is air cannot get out. So air cannot come in. That means the volume of the inspiratory volume decreases, and that's why all of the volumes decrease, because you cannot breathe this stuff in. But because PEF determines the amount of airflow you can expire, it doesn't change, because it doesn't the fibrosis doesn't affect that segment of the breathing. Okay, so to make the graph, we have to do a right shift. Easy way to remember restrictive causes right shift. Just remember this also, if you forget everything, and this probably will help you remember it. Restrictive causes right shift of the graph, of the flow volume curve. Okay, now we come back to the second graph. Now we have to understand how the second graph would change. We have the first, so we have the normal values, we have the normal one. Now in restrictive, one second. So in restrictive, what we have decided, what we have come to conclusion with is that the vital capacity decreases, right? Now we have to go back. Sorry. Vital capacity decreases, so we can see that you have to, in order for us to be able to draw this graph, you have to first decrease the vital capacity. And again, from the PEF, we have determined that the PEF doesn't change a lot. That means the force at which I need to expel the air out, it's not changed so much. So the forcible volume at forceful expiration at 1 second won't change so much. So that's why it's approximately same. So there will be a small decrease, but not so much. The biggest change would be in the vital capacity in restrictive. So to summarize both of them, in obstructive, we have the ratio in which there is we have the ratio in which the FEV is the main factor that drops. It goes down severely from 5 to 2 liters. In restrictive, the FEV doesn't change so much, but the forced vital capacity drops a lot, right? So based on this, we should be able to draw the graphs. Any questions until now? Guys, any question? I can move because then I'll move on from the restrictive and obstructive.
No, it's okay.
Okay, perfect. So just to add to the restrictive and obstructive, that we can have restrictive and obstructive based on the lung itself, or we can even have the chest wall impairment. If the chest wall has some form of, so there's scoliosis, there's fibrosis again in the chest wall, or if there's fractures, again, if the chest walls compliance decrease, you would have a graph similar to the restrictive, not the same, but similar to restrictive diseases. But again, you don't have to know the graph for it, so I didn't mention it. The second thing is, again, you can even have, so not only chest wall can be impaired, but you can have problems with respiratory muscles. Again, these can cause the formation of graphs that are similar to restrictive and obstructive, but they will not be the same. But you don't have to know them.
Basically, these are important for next year, for third year. But
Yes.
for physiology, just know obstructive and restrictive and how to do the graphs. And if you know how to do the graphs, then next year it will be so much easier.
Exactly, that's exactly it. So we discussed the part one where we used the we did this spirometry. We used all, so in spirometry we get this results. You guys should know how to interpret the result, and if asked, you should know how to form the graphs and how the graphs are formed. That means you have to show the thought process of how the diseases causes the changes in the volumes. It pathological changes in the lung, physical changes. These physical changes cause the changes of volumes in the lungs, and how these changes are different for restrictive and different for obstructive. And based on these changes, how the two graphs change. If you can explain this, you can answer any questions regarding spirometry. Do you have any questions regarding spirometry now? Okay, I'm going to move on to the PEF meter. I'm pretty sure you guys were able to use it when you had the practical. If you guys remember anything, do you remember what it looked like, or what did you guys do with this? Any idea, guys?
Yeah, so apparently they didn't do it.
Okay, you guys did not do. Okay, so the way this works is that you set this. So if I were to use it right now, I would set this thing to 0 first, and then I would breathe in as much as possible, and then put this in my mouth and expire to as fast as possible and to the complete expiration. Do you guys get it? So I take this, I put it to 0, and I put it in my mouth, and then before I put it in the mouth, I breathe in as much as possible, and I put it in my mouth and expire as fast and as much as possible. What it does is that, so remember initially when I said that this is used to figure out, just to have a basic idea if the person lung capacities or person's lung fall in the normal category. Well, this is what it does is that it measures the maximum speed of expiration. So it will only give you a general idea if the person has any kind of issue. And if the person does have any changes in his lungs, we can go further for spirometry. And the values it measures is in liters per minute. So suppose you get a value of 250 liters per minute. Now, how do we interpret the value? We can use, so again, there's a formula. No one has to remember the formula. You use this formula to find a predicted PEF. So you find a predicted PEF, and then you compare it with your value. And if your values fall between 80 to 120% of this predicted value, then you consider normal. If not, then you go for spirometry and figure out if it's restrictive or if it's obstructive. And then once you figure that out, if there's any other issues, there is another method of the helium. I don't have it in the presentation, but there's another method with helium that you can use to find the total lung capacity. And that's it. Do you guys, can you guys read the question and try to answer this? Are you guys, did you guys try?
So
Go ahead, yeah.
It's okay,
go,
Kira.
Lisa.
No, go.
So what is the answer? Any idea? What is the answer?
Kirari?
So I guess that it's a obstructive disease. So asthma and emphysema and yes. Bronchospasm. Yes, I guess this one too. And not the fibrosis. And I'm not sure about old age. Maybe
So why do you think it's not fibrosis?
because fibrosis it's restrictive. No?
Yeah, fibrosis is restrictive. Yes, yes.
So in case of restrictive disease, it doesn't change the how does it go, the peak expire flow, PE.
Yes, yes, PEF, yeah.
Yeah.
So what do you think is the final answer then?
So the my answer is A, B, C, and I'm not sure about E, old
Yes,
age.
it's
but
ABC. Yes,
yeah.
exactly.
Okay.
So
Okay.
exactly. Sorry, I didn't catch it. My internet cut off for a minute. That's right. Exactly. Okay, this is for me. Do you guys have any questions? I'll go back to the start, and if you have any questions, I can answer any questions.
Do you know the difference between FEV1 and peak expiratory flow?
Yes, do you guys know?
And can you show it on the graph, the flow cytometry graph? This one, this graph. Where is it?
Flow. This flow cytometry is this one. Flow volume.
Yes. What is the difference between FEV1 and peak expiratory flow? And where can you find it on the graph?
Mm.
Ace?
Stelios, your question is to find peak PEF.
Yes,
on
and
this graph.
On these graphs, yes.
Okay. Guys?
It's 7.
Sorry?
7.
Sound? I do not get
7.
7. Yes, exactly. PEF is 7. What about FEV1?
FEV1 is on the right graph.
Yeah, but is it
like-
possible to find it on this one?
No, because there is no.
There's no time.
Yeah.
Mm-hmm.
So.
But there is one way. If I were to say the graph is for a normal person, and we can calculate vital capacity, 80% of the vital capacity should be the FEV1. So what we can calculate from this is the vital capacity of the person, approximately 3.8. 80% of that 3.8 should be the FEV1. But again, this question, I don't know, Stelios, this is what you meant, or I don't know what you meant then.
Yes, this is one of the way, like estimation based on if it's a normal person, like you can estimate.
Yeah.
The other, which I don't remember exactly, is that the time is in the flow. So something with area, something with the air.
Yes. Okay. Yes, because flow is the volume.
It was a bit complicated, but they asked.
So let me think. Wait, flow is volume per second. It should be. Well, logically, it should be somewhere over here because that's the maximum. It's the what do you say? That's at that point you are expiring at the maximum flow, and then later on your flow starts to drop, and that's when you are unable to. So in the first second is when you have the maximum flow rate as well. So maybe
are you talking about the density of the dots? I remember that like frequency of the dots were telling us the time.
Which frequency of the
dots? Like the density of the dots. Like,
yes, I remember. I think
because,
it was.
It Professor Kubichak asked us.
I don't know, catch that. What do you mean?
The density of the dots, like the frequency of the dots, are telling us the time.
Yes.
Right.
So basically, the machine is collecting one dot every, I don't know, like millisecond or something. So you actually measure the dots. Wow, you remember.
So based on that, you can calculate FEV1.
It was something Professor Edward was explaining, but
Okay.
I don't know if he will ask this in the exam. But he asked it in all the practicals. He asked this question: how can you find FEV1 on this graph? And the answer was the machine. Now it's digital. It collects all the time. But before it was analog, so it was making a dot every, I don't know, 1,000.
Okay. I did not know this. It's good you guys remember this. Yeah.
Just know the density of the dots. If they ask in the exam, they say, yeah, based on the density of the dots. You don't need to explain it further.
Yeah, I would just go for approximation. I just approximated it. I just used, if it was a normal person, then I can approximate it. Okay, guys, any other questions?
So in the final, if the examiner asked us about the FEV1 in the left graph, we just answer density of the
dots.
dots.
I think Ace and Stelios knows this. I actually do not know. Do you guys, can you, one of you guys explain this, if that's what you want?
Kirari, if you remember, Edouard, I remember in the lab, he went at the end and he asked this question, and then we spent, I don't know, like 40 minutes on it. And he made us draw something on the board. Remember, he called someone, and after he drew something, I remember I have in my notes the drawing. And then he said that to find the time is just the frequency of the dots.
Okay, well, remember the frequency of the dots, then frequency and density of the dots. I guess that should answer the question.
Okay, thank you.
No
and
problem.
just, you know, the space between the dots, like how fast is it, or that's all. Just this.
Okay, thank you, Rita.
No problem. Any other question?
So that's it, I think.
Okay, guys.
We're going to send the presentation. Me and Panchu will merge it into one and put it on the physiology class.
Thank you very much.
No problem. Thank you. Bye-bye.
Thank you so much.
No problem.
Bye.